Hello friends, how are you all? I hope you all are doing fine. In this video, we will explore Seurat. Okay, Seurat is a popular R package which is widely used to do the downstream analysis of single cell data. Okay, so this is is used after the CellRanger. So if you remember in the last session we talked about CellRanger where we saw how we used means how we converted the raw FASTQ file into the count data. So now the Seurat comes after it where we can feed the output from CellRanger and do the lot many downstream analysis. Okay. So far what we have learned, yes, we talked about how single cell technology works and we talked about CellRanger in details. So in this video mainly we will talk about how to process the raw count data and then analyze using the Seurat pipeline. So Seurat is a popular pipeline and it has multiple steps in it. Okay, so like once how to load the data into Seurat, then we do the QC and pre-processing followed by normalization, then we do the scaling, then we do dimension reduction like PC analysis kind of things, then we cluster the cells and later on we annotate each cell subtypes. Okay, so you can see that there are multiple steps are there in the pipeline and each of the steps are very important. So we need to understand why we are doing that and how we are doing it. So whenever you are learning, right, we need to learn about two things like why we are doing it and how we are doing it. So that's where I will be splitting this Seurat pipeline into multiple video where so that we can talk each of these concepts in details. Okay. So particularly this video I am focusing on first load the data into Seurat and we will see how Seurat stores the single cell data. Okay. So let's start. So this is the overview of the pipeline. Like so we get the BCL files. So we use MK FastQ to the CellRangers command MK FastQ to convert the BCL files to FastQ files. So this is the raw data. Then we use simply the count command to convert this FastQ into the read count data. So this is the final data from the CellRanger. So this is stored in basically three files. So one the barcode file, the feature file and the matrix file. So this is the count matrix, right? Each column corresponds to cell or barcode while each row corresponds to gene or features. So that's where you need to know the barcode or cell level information which is stored in the barcode file. So the feature file will contain the information about the each of these features or genes, whereas this matrix.mtx file will store the count data. So that's why we need these three files. So, and these three files are usually stored into a single folder and we will be uploading this folder into the Seurat directly. Okay. Now this is the pipeline. I will just give you an overview of the pipeline. So first we load the data to Seurat, then we need to do some QC and quality check and we do the pre-processing, so where we remove low quality cells. So once we obtain high quality good quality data, then we do the normalization and then we do the scaling. We'll talk about why we do this. So once we have normalized our data, then ultimately we do the dimension reduction, then we do the clustering and we will be using different visualization method like TSNE UMAP, and then we will identify different biomarkers within each of these clusters, and then we usually do annotate each of the cell subtype. So that is the overview of the Seurat pipeline. So we will be focusing in this video on loading the data to Seurat package. Okay. So good. So now in Seurat we will see like there are two functions in it. So before that, let's start with installation of the do the hands-on. So this is the Seurat website. You can click on the install button web page. So the installation is very simple. Install.packages Seurat. I have already installed in my system, and you can once it is installed, we can load the library using this library Seurat. Okay. So now in this example, what we will be doing, we will be using the same tutorial files given by the Seurat team. So we will be following this tutorial, guided tutorial, where 2700 cells PBMC samples are there. So you can see that this is a dataset of peripheral blood mononuclear cells, and there are around 2700 single cells are there, and they were sequenced on Illumina NextSeq platform, NextSeq 500, and the raw data you can download it from here. So by clicking here you can download it. So I have already downloaded this file, this tar file, and I have extracted it. So now if you go inside you can see that this is the output from the CellRanger. Okay, and as I stated, like, so we'll have the barcode file, and the gene file will contain, or the feature file will contain the gene information, and the count data is stored in this matrix file. So we will be giving this folder directly as the input to Seurat. Okay, so now coming to here, so there are two functions in the Seurat package. Okay, so one is read 10x function, or then create Seurat object. So basically this function, where it will take the these three files as input, and it will merge everything into a count matrix. Okay, so count matrix will look like this. Okay, so this function will take, read 10x function will take these files and generate the count matrix, and then we use this count matrix and store in the form of a Seurat object. Okay, so two steps are needed. And if we can also use the binary file, if you remember, like the CellRanger also outputs the same information in a binary file, HDF file. So if we are loading this file, we can use this function instead of this, which will convert the same this data to a count matrix. Then we can use the create Seurat object function to convert the matrix to the object. Okay, so first we will be converting into matrix, then we'll use the matrix to generate the count object. Okay, so now let's start the practical. Okay, we open the R Studio. Okay, so I'm just setting my working directory. Okay, and I'm loading the packages. Okay, so I will be also using this dplyr package. So first you need to give where the output of the CellRanger is located. You see my working directory is here, right, Seurat. So here, and within that I have my data is inside this folder, and then within the filtered gene, then HG19. Okay, so that's where I'm giving the path in the current directory. Now this is the folder, and then this is the folder, and HG19. So this is where my data is. Okay, so I will be creating this variable. So first we'll be using this function read 10x. Okay, so the first argument is data.directory, where we will giving the path. Okay, so I'm giving the path I created with this variable. Okay, and let's store this into this variable called pbmc.data. Okay, so now what will happen, it will now load those three files and automatically convert them into a count data. Okay, so we can use this variable. Okay, since it is a matrix, right, count matrix, so you can use the same syntax like as the before. Okay, so okay, let me just organize them. Okay, so this is the command. You can see that this is a sparse matrix. Okay, this is not an ordinary matrix. Instead, it is a reduced representation of matrix, which basically saves a lot of memory. Okay, sparse matrix, and you can see that instead of 0, we are we are having dots. Dot means that gene is either not detected, or it is gene count is or it is a dropout. Okay, so basically we are it shows sparse way of storing the data. Okay, and yeah, so this is a matrix kind of thing. So now in the next step, so now we got the count data, right, using this read 10x function. So we will be now using the second function create Seurat object. Okay, so this, the first argument is count, so we will be giving the count data here, pbmc.data. This is my count matrix. Then you can give some project name. Okay, so let's give this name, and then we give minimum number of cells and minimum number of features. So these two parameters we can give. So what does it means, like we are giving some filtering here. So first filter is, let's say, if a cell does not have this many genes detected, we can discard those cells. So a cells to be included, they must have 200, at least minimum 200 number of genes should be expressed. Okay, then only we will consider them as a cell, and we will include them. Similarly, if a gene is not expressed in 3 or more cell, then discard them. So at least a gene has to be expressed in 3 cells, then only include. So using these two criteria, right, so using this two criteria, we are now loading it. Okay, so basically this will initially do some filtering. Now if this is the Seurat object, so if you type pbmc, you say that it will show it is an object of class Seurat, and it has how many features or genes are there? 13,714 genes are there across this many samples or this many cells, right? So if you remember, right, we have the count matrix, right? So where columns are cell and rows are this thing, right? So that's where if you type dimension, right, it will tell us like this many row and this many columns are there. Okay, so we can use call names of, let's say, pbmc. I will just show you few, columns. So you see that, this is the column names, these are the cell barcode, first barcode, this is the second barcode, this is the third barcode, like that. Similarly, row names, okay, of will give us what, the gene name or the feature name, right? So this is the first feature, this is the second feature, this is the third feature, like that. So, and there are this many genes and this many columns are there. So now one more important thing, you can see that this is one essay. Okay, so now that's a very important concept here. Okay, we'll see the Seurat object. So now when we do a single cell experiment, right, we can have multiple experiment. Okay, and the results of all experiment can be analyzed together. So there then we can have multiple assay within a one object. Okay, so so like we can, for example, this is the Seurat object, right? So within that you can have multiple assay, SA1, SA2, SA3, like that. Okay, so in this example we had only one assay, and that assay name is RNA because this was a gene expression data. Okay, so now coming to Seurat object, right? Okay, so if you just expand this PBMC, right, so you will see that first it has different slots. Okay, so now it is an S4 object, and and that object has many components, or we generally call them slots. Okay, and you see that, and slots are usually always starts with at the rate. So you can see that a Seurat object will have different slot. The first one is SA. In this slot or in this component, basically this stores a list of SA. So you can consider SA as one experiment. Okay, like that, very layman term. Okay, so you can store multiple experiment results or multiple SA within this slot. Okay, so now this component or this slot will contain the metadata about each cell. Means what is that cell, any metadata you can store about the cell. And the third, there are some others are there, but important one is also the reductions. So this slot can store all the PCA results. Okay, similarly, clustering results all can be stored over graphs, like that. Okay, so we can see that there are different slots are there. So PBMC, the first one is SA, and if you go down, you can see that at the rate metadata is one slot, active SA, and graph neighbors reduction, these are different slots. As I state, PC analysis results will be stored here. When we do the clustering, right, cell clustering, all the results will be stored here. So now you can see that it is a blank canvas kind of thing. Okay, everything is empty, but as and when do the analysis, right, so all the results will be stored in this single object. So we don't have to create, multiple variables, like, PCA results store in PCA variable, clustering results store in cluster variable. No need to do that. Just the single object, and there we can store all the results. So that's the beauty of, object oriented programming in R. Okay, as for objects. Okay, so clear. So to just, we will see how to access them, but now you can see that a Seurat object has multiple slots, and the first one, important one, is the SA slot, and the SA slot is also within that different slots are there. So this SA, for example, one experiment will store, within that you have three different slots. First one, the count slot, this will store the raw count data. Okay, so now when we do the downstream analysis, normalization, the normalization results will be stored in this slot. When we do the scaling, right, the scaled results will be stored in this slot. Okay, so now to come here, you see that this is PBMC object. Now the SA slot, and within that it's a list of SA. So currently in this example we have one SA, so this is a list of one element. The first one is RNA, and within that, this itself is a class SA. That means it has different slots. The first slot is the count, which is a matrix, and then we have a slot data, and then we have scale dot data. And let's say we have, variable dot features. So this will store, when we do the variable feature analysis, those results will be stored here. Okay, so indirectly what I mean to say, like, the single object can store all the results. Okay, now we will see how to explore each of these component. Okay, very simple. So you start with PBMC. Okay, now if you want to, go over here, right, so you can have to use this at the rate symbols, right, because before SA we have at the rate symbols. So the moment I type at the rate, then it will show us all the slots within it. Okay, so now let's say first let's go to the first slot, that is SA slot, and this will store what, a list of SA, right? So now then we will type dollar because it's a list, so and the dollar is there, and the first the moment I type dollar, it will list all the assay within it. So now in this example we have one assay, so so that's RNA. So now this is itself is another class. If you see that this is what assay data, okay, and the class is what assay. So now this within that, we have different slots. So again I have to type at the rate. So the moment I type at the rate, it will show all the slots within this class. So the first count is a count matrix. So I am just showing you three, row, three columns. Now if we have normalized data can be stored here, so I can use the second slot at the rate, then data slot, okay. So 3 row 3 columns like this, okay. So this way you can access all the slots within this, so you can navigate like this, at the rate, then dollar, then at the rate, like this, okay. So now this is about the SA slot. So now very important slot, the second one is metadata. So this contains the metadata about the cells, okay. So now how to reach that PBMC at the rate, then metadata. So basically this is a data frame, okay. So you can see that metadata is it's a data frame. So that's where we can use head to see the content. So now you can see that each row is what now, this data frame, each row is one cell, and the annotation about the cell is present, okay. So now in this example you can see that this is the first cell, and in this cell number of RNA molecule detected is 2419, and those many RNA map to 779 genes, okay. Similarly in the second cell we have this many RNA molecule detected, and those map to this many genes. Third one 3000, that many RNA molecule detected, and they map to this many genes. So now the dimension, if you check, right, this will be around what, in this 2700 cells, and 3 columns are there. That means currently we have 3 metadata about the cell, okay. So we can use this at the rate notation also, and there is second notation we can use is double bracket. We can use the double bracket notation as well to get the 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 fetching the metadata. Got it, right? So yeah, so one is PBMC at the rate, we can use SA, and then most important is the metadata. So these two slots are important, and yeah, and the third approach is like we can also use this get SA data function, another way to fetch accessing, okay. So we can use this function where we pass the object and which slot you want to fetch. So let's say in this example I want to fetch the count slot, so I can say this the object and what data I want, like that, okay. So that is all about how Seurat stores the object, stores the single cell data in the form of single object. It is very important, I had to explain, because once you understand the familiarity with this function, so you can play around it, right. So that's why it is very important to know the Seurat object. Oh, I think that's all about in this video. In the next video, we will talk about the next step in the Seurat pipeline, where we will do the QC and pre-processing. So we will talk about why. Means why we are doing QC and why to do the pre-processing and how it is important. Okay. So I think this. I hope this video was useful, and if yes, so please press the like button, and if you have not yet subscribed to my channel, requesting you to subscribe it, and don't forget to press the bell icon, because when I post a new video you will get a notification. Okay, thank you so much for the support. Bye bye, see you in the next video. Bye.